We are live now, sir.
Good. Yeah. So, good evening, all of you. I'm sure all of you are enjoying the course, finding it useful for your studies. And if there are questions and queries, then you may use the chat box that is on the left side, on the right side of the YouTube, and then you can write. Then I'll be able to answer. So to begin with, I'll take up the questions that are already posted in the portal. I think 3 of the questions were on the assignment. In fact, one of the assignments were in the DNA sequencing gel. We asked you to identify the exact sequence of the template DNA. I think that question, there are multiple queries, and since the so-called correct answer given on the portal was not matching with the answer that is given in the answer of the assignment when it was released, and there was an error, so that will be fixed. What is given at the end of the assignment, the worked out answers are correct. So those who have opted for the correct option, you will get full marks. So you have to wait for it. That will happen. You don't need to worry about it. There was one question on week 2 assignment, question number 3 and 4. The 3 was on the restriction map. Fourth one was on the the penetrance, meaning how many asymptomatic carriers in the pedigree. I think that the restriction map is something that we'll work out and show you on the portal. But it is, when you run a agarose gel for any digester fragment, you have to understand if there are 2 fragments are of the same size, it's going to migrate at the same position. Therefore, it would look like single fragment. That is what that would happen, and that is what's sort of tested here. Because if you see there, between EcoR1, if there is a BAMH1 site in between, then if you cut it, then each fragment is identical, so it will run as a single fragment. Likewise, if you digest only with EcoR1, you are going to have 2 kb fragments. Again, that would run together. Therefore, it would look like single fragment. So this is what one should consider while doing the mapping. So in other words, now we have given the map and we tell you what kind of a results you are getting. But basically, you make the map based on the fragment size that you are getting. Say, for example, if you are given a DNA fragment for which you don't know the sequence, and you have to make the maps, basically you digest the DNA with a given enzyme, 1 or 2 enzymes separately, and then look at what are the size fragments that it produces, and then you do a combination of digestions, and that is how you do the positions, restriction maps you create. So you have to keep that in mind. In this case, when you say EcoR1, if you digest 4 and 2, some of it is not making into the total. If you see that it says 8 kb is the size of the plasmid, but if you digest with EcoR1, it is 4 and 2. That means that one of the fragments should be double, and it cannot be 4, because 4, if you have 2 fragments, that alone becomes 8 kb. So it has to be the 2 kb fragment that should be 2 in number. Therefore, that becomes 4, and then you have another fragment 4. And BAMH1, of course, what is the question here? Which one of the options given below identifies the correct number of DNA fragments, right? Yeah, that is what it is asking here. So I think there was an error here, or what? Yeah, there was an error here. Yeah, there was an error. So we'll answer you. There is an error here. The other question is calculate the minimum number of carriers for the mutation in the pedigree shown below for a dominant phenotype with incomplete penetrance. And if you see here, then in each generations, you will find For example, the father of the son affected in the third generation, the father is not affected, yet he transmitted the mutant allele to the next generation. So that is what's shown in the worked out assignment. You can see there is a red arrow. That means that person is a carrier, must be a carrier. Likewise, the fourth generation, the great grandson, his father also should be carrying the mutation, the mutant allele, but he is asymptomatic. So minimum number of carriers should be at least two. It could be, that is what that you can predict. So that is the answer number two. Um, so these are the questions that were there in the portal. If there are other questions, we will look into that. Sanjay has written how to identify nonsense mutation. Yes, a nonsense mutation. Okay. Well, it when you sequence any gene, you have to what is the open reading frame, and because the open reading frame starts with the start codon, which is ATG, and then from there you have the triplets, that is the codon, right? If any change that in a codon that makes it nonsense, meaning a stop codon, that becomes a nonsense, because in codon table, if you look at what are the codons that really translate into a stop codon, right? So such changes, we know that this is nonsense. It's as simple as that. So you may post your questions on the chat box, so we'll be able to answer. We'll wait for your questions. So when you write, we'll do. Okay, Sanjay has asked the question again that can you please explain the same, that is nonsense mutation, with an example? That is what his question is. There are this stop codon could be, for example, UGA, or it could be UWA or UAG. These are some the codons that gives a message during translation that translation has to stop there, right? Now, if you look at the codon table, you'll find that UGA is a stop codon, but UGG codes for an amino acid, right? You assume that in the UGG, that G gets lost base in the codon, gets converted into A because of a mutation. Now, instead of the amino acid, the RNA resulting from that particular gene would give a signal that the translation has to stop there. This is one of the ways by which a nonsense mutation can happen. Or for example, UAA or UAG, all these, it can be, for example, UAA, UCA is serine. So if C gets converted into A, again it becomes a stop codon. So it depends on the kind of combinations, what kind of changes happen, in which position happens. And therefore, a codon that otherwise codes for an amino acid now gives a message that it is a stop codon. So that is nonsense mutation. Is it possible to trace and find out, is there any recurrence of breast cancer by genetic analysis? Uh, yes, I mean, I will not restrict my answer to a breast cancer or anything, but there are cancers which are because of somatic mutations, meaning mutations in the somatic tissue, which is other than germ cells, and therefore these are mutations that happened after you are born, after you are grown up. In a given tissue, there could be mutation that leads to, for example, cancer is a possibility. Some of them, of course, are germline, mutations as well. But when you talk about recurrence mutation, is that you have done a chemotherapy to remove or kill all those cells that carried the mutation, and people do this translocation, what we call as Philadelphia chromosome in case of leukemia, right? So that happens because of that. There is a fusion of two different chromosomes. As a result, a gene gets activated, and then you end up having leukemia. And there, the patient undergo chemotherapy, and then, in fact, people do that kind of a mutation screen to see whether the therapy was effective. And one can periodically go and do this diagnosis later to see whether there is a recurrence. So yeah, that kind of an approach can be used genetic if we know for sure uh, the cause for that disease or cancer is genetic in origin, and we know the what is the gene, and certainly we can do the screening. Knock-in model. Knock-in model is see knockout. When you say knockout, is you basically inactivate a gene. That is a knockout, right? So you delete part of the gene, either the first exon or exon that is coding for a functional domain of a protein. You delete, that results in loss of function of the gene. When you talk about knock-in model, you basically you create a particular mutation, which is, say, for example, known in a patient, like a missense mutation. And you want to model that missense mutation in a mouse model, then we create what is called as knock-in model, wherein it is not necessarily that the mutation that you are creating is inactivating the gene, but it results in the same kind of a change that you see in the human, for example, or the kind of mutation that you want, which could be simply, replacing one base. Therefore, the codon changes; it becomes a missense mutation, and then now you allow the animal to to grow or look at the phenotype as to what effect this particular mutation has on the physiology, development, behavior, whatever parameters that you are studying. So that is the knock-in model. The approaches that are used for this is very similar to what we use in knockout approach. Basically, you have the construct which has got the desired region of the gene, then you create a mutation therein, and then you allow it to undergo recombination, and then replace that. You transfer that into embryonic stem cells, and then the embryonic stem cells in which it got incorporated, use that to create the chimeric mouse and go on doing it. So the process is very similar, except that it is not necessarily inactivating the gene, but whatever changes that brings in that mutation. So that is what it is knocking. There was a question on week 3 assignment, question number 5. It's about the week 3 assignments. We have released the answers as well. The question is about, so there's a schematic given for the genomic segment, and the gene targeting construct is being readied for generating a knockout mice. Question is, identify the segment that is likely to be integrated into the genome if the construct integrates into the genome via homologous recombination. That's the question. So in the targeting construct, you have a toxin, we have the flanking region, then you have neomycin resistant coding sequence and another flanking region on the right side. So the question was asked is, which likely to be integrated into the genome? So the answer given is neoresistance, because the exon and the neomycin, they don't have homologous sequence, so it cannot recombine there. But the two flanking regions are, identical sequences, so there will be a recombination between them. If recombination happens on both sides, both the flanking sequences, then the neomycin resistance sequence would replace exon. So that is what is expected. So that is what's shown in the schematic. Yeah. If you have more questions, you can post. Maybe we'll wait for a few more minutes. And in case there are no questions, then we may wind up the live session. Somebody is asking, is it enough if we learn the video lectures alone for the final exam? When you really want to gain knowledge, there is nothing called enough. As much as you read, as much you understand, right? The lectures are always, you should consider these as something that is introductory, which sort of introduce you to the concept. And, therefore that serves as a launch pad for you to go to the depth of the topic. That is the reason why we have these lectures. And it certainly helps. We have given you the links for additional reading material. And if you read that, that is good enough. Whatever reading material that we are given, the links and so on, they are not very beyond the topics that is covered. So it is very much relevant to the topics that are covered in the lecture. So I would suggest that you go through them. In addition to the video lectures, you certainly read some of the links that we give for each topic. And that gives you good understanding on the concept, because the examples that are given in an exam may not be exactly the same kind of examples that are shown in the lectures. It can be somewhat different. So if you have a better understanding, you will be able to answer it better. Somebody is saying, where can we find reading material? You're adding them in the announcement. I think it is in the announcement, along with the announcement when the lectures are, released. Along with the announcement, the links are given, so you can go and check your portal, course portal. It will be there. Yeah, in addition to the links, also lecture notes are also given, but I think we have given links to some of the freely available reading materials, so in the internet. That is also posted, so you can go and look at the announcements. The transcript is different. Transcript is, basically a text version of the lecture. That is one, but we also have, given the links. No, see, so one of the question is give us some worksheet for restriction mapping, pedigree problem. There are plenty of them on the internet. Just simply you type a Google search saying pedigree analysis problems. You'll find lots of problems that is available. You certainly can work it out. Whether it's a restriction mapping, pedigree problem, all these are available on the internet. You can check it out. I did not mean transcript. What I meant was links that are given for. So somebody has responded already. Um, there are links given for the topics that I think you should go and look at your inbox, or you go and look at your course portal. It's all given there. I mean, I think you need to really look at that. Well, I mean, Sanjay, you may post it again in the discussion forum. We will link you there. It is available. Now, I will not give you mail ID and all. You post it in the forum, and one of your own, like you can see, Ujjwal has written that the link is there in every mail that is sent to us each week of the course. So I think you are not looking at carefully. Yeah. You post this query in the discussion forum, you will get the answer. Yeah. So if there are no more questions, then we may end this session. I don't see questions coming. Tips is very simple. You read well, you will do well. Yeah. For any exam you want to take, the only advice is that you study well, try to understand the concept. You certainly can do well in your exam. So then I think we can end this session because there are no more questions. Yeah, then I'll end the session, and I wish you all all the best for the exam and do well. And maybe we can see again in some other course. Good luck to you all.